Now let's start this exam. Even if you solved it before, we can discuss it together. Now we are in November 2023, paper 22. Let's jump straight into it. During interphase and mitosis of the cell cycle, the chromosomes within a cell go through a number of changes. Each
We cannot
chromosome...
see the screen.
Oh. Thank you for telling me. 1 minute. Is this better? Now you can see the screen. Can you see the screen?
Yes.
Yes.
Okay. We are in number 1. Each chromosome is composed of DNA complexed with proteins. In interphase, individual chromosomes are too diffuse, they're too long and thin to be visible using a microscope. In this stage, the chromosomal material is known as chromatin. State the term used to describe the proteins that are complex with DNA and form part of this protein. What's the protein, Noor, that is associated with the DNA, that the DNA coils around?
The name of the
Huh? What?
the question asks the name of the
I cannot hear you. We said that the DNA strands, they coil around a certain structure because this structure will help in the supercoiling and the packing of the DNA.
histone.
Yes, histone proteins. Okay. Give me a second. Yeah, 1 minute. What is this? So this is histone. Okay, when viewed through a microscope, a chromosome is most clearly visible during metaphase. Complete figure 1.1 to produce a labeled diagram of the metaphase stage of mitosis in an animal cell with 2 chromosomes. And be careful, this is a labeled diagram. So how will you position the chromosomes in metaphase, Mariam?
In metaphase, the chromosomes align at the middle part, also known as the equator.
Very good. Okay, you will now draw 2 chromosomes. How will you draw them?
They are stuck to each other with
Something like this.
yes.
Okay, so each one of them is a chromosome made up of 2 sister chromatids because they want you to label. So label everything that you know. These will be considered sister chromatids. The whole structure is 1 chromosome, and this meeting point node, what is it?
Centromere.
Yes, these are the centromere. They have another name they call kinetochore, but the centromere is more common. So the centromere. These centromeres, they are attached to what?
Spindle fibers.
Spindle fibers.
Correct. And those spindle fibers are originating from where? They're coming from where?
The poles of the sun.
They're coming from an organelle. This organelle produces the spindle fibers. What's the name of this organelle? Now, this organelle is at opposite poles, correct? This organelle is the centrosome or the centriole. We draw it like this, okay? So you will label this as the centriole. These are the spindle fibers. Okay. This is the chromosome, 2 sister chromatids, and they line up at the middle or the equator. What is the middle part called?
Centromere.
The
لا
middle part of the cell
equator.
plate, metaphase plate, or the spindle equator, or the equator only, whatever. So or metaphase plate? Metaphase plate is more common. Metaphase plate. Okay, so everything here is labeled. Outline the changes that occur to the structure and behavior of a chromosome from the start of the synthesis phase to the end of the interphase and during the prophase of mitosis. So here you need to outline what is happening to the chromosomes from the start in the synthesis phase to the end of the interphase and during the prophase of mitosis. Who will remind me what happens in the synthesis phase?
DNA replicates.
Yes. Okay. What will you write here?
The DNA replicates.
Okay, what's the name of the replication? The DNA replicates. Very good. Not only the DNA, by the way, the DNA and the centrioles or the centrosome, they replicate. And the DNA replication is called, it has a name. I just want the name.
Semi-conservative replication.
Yes, the semi-conservative, just write it down. Semi-conservative DNA replication. Now, what happens or what comes after the synthesis phase? We have the interphase, it's G1, and then the synthesis, and then the G2. What happens in the G2 phase? We said the synthesis replication, correct. And then what happens in the G2 phase?
More growth.
Yes, very good. More growth of the DNA. And something very important happens in the G2 phase.
Errors are checked in the synthesis
Very
phase to be
good. Yes, the DNA replication is checked. Any errors are checked. And if we can fix them, we can fix them. Good. So clearly, don't mix things up. If you mix things up, you lose marks. In the synthesis phase, the DNA replication, semi-conservative replication of the DNA, and also the centrosomes replicate, if you remember it. The G2 phase, I have more growth of the cell, making of proteins and growth, and we check the replication of the DNA. Now, during the prophase of mitosis, we said prophase is divided into two parts. Who remembers them? And in total, there are 5 things happens in prophase. Quickly.
early and late prophase.
Yes, we have early prophase. And we have, so this is a straightforward question. What happens in early prophase?
The nuclear membrane and the nucleolus break down.
No, this is in the late. Only one thing happens in the early prophase.
The chromosomes, they condense, they shorten
Yes,
and
very
double.
good. Good. Yes, the condensation of the chromosomes. So the chromosomes, they condense, they become shorter and thicker. That's correct. This is one. And then late prophase, 4 things happen. So in total, 5 things happen. Like you said, Mariam, what happens in the late prophase?
The nucleolus and the nuclear membrane disappear.
Very good. 2 things disappear. The nucleolus and the nuclear membrane, they disappear. And what appears?
The spindle fibers.
Very good, the spindle fibers. And who produces the spindle fibers?
The centrioles.
Very good. The centrioles, they start moving to opposite poles of the cell, and they produce the spindle fibers, which hold the chromosomes at the centromere. So it's very easy, okay? It's an easy question, straightforward question, but it's very clear. You have to be very clear what happens exactly at every phase: synthesis, DNA replication, and then the G2, because they said from synthesis to the end of interphase. So don't forget the G2. Okay, don't mention the G1 because they said from synthesis. And what happens in prophase? Very easy and straightforward question. Keratin is the structural protein in feathers of birds. Keratin polypeptides are composed of a high proportion of cysteine amino acids, which have sulfur-containing R groups. Keratin polypeptides form filaments, and the 2 main types of keratin in feathers are the alpha keratin, which consists of many alpha helices, and beta keratin, consisting of many beta pleats. Keratin can be classified as alpha keratin or beta keratin based on a study of protein structure. Suggest the level of protein structure used to classify a protein as alpha or beta.
Secondary.
Yes, I want to see who do we have with us online. Give me a second. Okay, hello, Adam. I want you to participate, please. Yes, the second level of protein, or this level of protein is the secondary, correct, because of the alpha and the beta. B, Adam. Protease enzymes hydrolyze proteins. Many proteases are able to hydrolyze more than one type of protein. Suggest why it is possible for a protease to act on different types of protein. This is a suggest question, but it's easy. Can you give me an answer, Adam?
Because of the induced fit mechanism.
Okay, now discuss.
Excuse me?
Okay, discuss what will you say in this part, in the induced fit part.
Because the R groups in the enzyme active site alter themselves so that they can fit around the substrate shape.
You are correct. The same substrate or different types of substrate?
Different types of substrates.
Yes, the active site can fit, okay, around different types of proteins, different types of substrate. It can move around a little bit. You're right. Induced fit, that is going to be acceptable. And it can be complementary to more than one substrate. Okay, I don't need to look at my screen, that's too easy. Proteases known as keratinases vary in the extent to which they can hydrolyze keratin. Feathers are not easily degraded. They're not easily broken down because keratin is a very stable protein. Suggest features of keratin structure that contribute to its stability. The first thing you will do is that you will look at the number of marks, 3 marks. So I want you to suggest why keratin is stable. I want 3 suggestions. They're telling you it's very stable, it's not easily broken down. Why?
It is insoluble.
Correct. Number 1, it has to be insoluble.
No polar ions.
You need no polar, correct. If it is insoluble, no polar or no hydrophilic R groups, you mean?
Yes.
The R groups are hydrophobic, yes?
Yes.
Okay, I want 2 more.
Contains a very high
Remember,
number of hydrogen bonds.
excuse me,
Contains a high number of hydrogen bonds.
only hydrogen,
Covalent bonds.
only.
Peptide bonds.
All bonds, all bonds. They said that these keratinases, they will here, these keratin polypeptides, they form filaments. So they had a primary structure and a secondary structure, which is the alpha helix and the beta keratin, and then they form and then they join to form filaments. So they will form a tertiary or even a quaternary structure. So they contain all the types of bonds that we studied, which are what? Include which bonds?
Ionic, disulfide,
Yes,
peptide, hydrogen, well, hydrophobic interactions.
hydrophobic interactions and hydrogen. Remember the high bonds? Leave the peptide. The peptide is the primary structure. In the secondary structure, we have the alpha and we have the beta. In the tertiary and the quaternary structure, we have the remaining bonds that join by the R groups. These bonds, they give stability and make them insoluble and make them strong, which are the hard bonds, and you can discuss them. The first H is the hydrogen, the hydrophobic interactions between the hydrophobic R groups, the disulfide bonds, they already said that they contain cysteine amino acids, and the disulfide bonds are very strong bonds. And you have the ionic bonds between the charged R groups. Okay, so they include lots of bonds, and you will discuss them that make them stable. Also, we studied 2 types of proteins, fibrous and globular. Do you think that this type of protein is fibrous or globular, the keratin?
Fibrous.
Fibrous.
Fibrous, because it was the fibrous that was insoluble, the globular, it was soluble. And suggest means that you can write a sentence starting, it may. We don't know. We did not study this type of protein. So it might have crosslinks. It might be twisted like the collagen. It might run in so many different directions. It might join, or these filaments, they can join together more and they form fibers. All this will be accepted. They will tell you definitely in the mark scheme any valid point. I should look at the mark scheme for this question. So let's say if we said 3 points, we said insoluble, and we said that the polar groups, the R groups will be non-polar hydrophobic. And if we mention all the bonds, the hydrogen and the hydrophobic and the disulfide and the ionic, we will definitely take 2 or 3 marks in this part. And also, if we mention it is a fibrous protein, not a globular protein, we will take a mark for that. So you will definitely, with what we said, we will take the 3 out of the 3. We will look at the mark scheme, so we'll see if we have any other ideas. Okay, this is the exam itself. The mark scheme will give us ideas. Always look at the mark scheme. This was which question? I think it was 2C. No. Is this the right exam? No, this is not the right exam. 2023, round 7. Paper, no, this is paper 23. So I think we're 22. Okay. Assume sequential interface to protein. We said that. Oh, here. 2C. Okay, any 3 from. Let's see if we will have, or we will take the 3 marks or not. Fibers, we said fibers, insoluble, does not dissolve or does not, we said that. High proportion of disulfide bonds, we said that. Medium said covalent bonds, high proportion of many hydrogen bonds, hydrophobic, all the bonds. We mentioned all the bonds, correct. Many amino acids with hydrophobic R groups, tight close packing of the chains. We said it may have close packing, it may have filaments, it may run in so many different directions, it may have crosslinks, anything that will give them strength. So we said that also. Any valid point arranged so the peptide bonds are not accessible. Part of the molecule needing to bind to active site not accessible. Many amino acids with small R groups. We talked about the R groups. So we will definitely take 3 out of 3. I don't think the mark scheme gave us any extra ideas. Okay, very good. Graphs. I hate graphs. Keratinases are used to degrade the large quantities of waste feathers from chickens and turkeys that are processed in the food industry. So we are talking about this enzyme. The products of feather degradation can be used in animal feed. Scientists investigated whether 3 different keratinases were suitable as industrial enzymes. These enzymes were extracted from 3 different soil bacteria. So we are studying 3 different enzymes. The effects of temperature and pH on the activity of each enzyme were investigated, and the results are in the figure. You have to always look at the figure. You have one graph for temperature on the x-axis and the relative activity on the y-axis, and you have a key. The circle is one enzyme, and the X is another enzyme, and circles inside the circle is a third enzyme. And you have another graph for pH. Of course, they will ask you to compare and describe and explain. To degrade feather waste from industry, it is an advantage to use keratinases that show at least 60% relative activity in conditions where temperature and pH can vary. Table 2.1 shows for each keratinase the working range of temperature and pH where at least 60% relative activity is obtained. Use figure 2.1 and figure 2.2 to complete table 2.1 and use the completed table 2. Name the keratinase that has the widest working range of temperature and name the keratinase that has the widest working range of pH. So, keratinase K12. The temperature range at which there is at least 60% is from 41 to 63. pH range with at least 60% relative activity. Okay, you will go to which graph? Temperature or pH to get the number here? Adham.
pH.
You will go to pH, very good. And you will look at the key because I'm looking at keratinase key 12. So what will I do? I have to just be very careful in these questions and read them carefully. I'll go to the pH graph. I'm going to go to the relative activity 60, and I want K12, which is the solid circle. I'm going to go to this graph and I will look at the pH range. The pH range is from here to here. Do you understand how I got it? This
Yes.
is this is 60. So I'll go to the pH range from here to here. From less than 7, nearly 6.1, 2, 3, 4, 5, 6, 7, 8, 6.8 to 9. This is the pH range at relative activity of 60% for the stated enzyme. We said 6.7 to 9. Let's see if we are right. 6.7, 6.8 to 9. Yes, around 8.4. Okay, let's go back. It's clearly 9 here. Let's just make sure this is the correct enzyme. They said which enzyme? K12. K12 is a solid circle. We'll go to pH. 60. You need to get a ruler. 60. Okay. This graph. See, it's confusing. You will go to this graph until it stops you. So the value from here until, and this is the graph. Yes. So it is 6.7, 6.8 until 8.1, 2, 3, 4. 8.4. Do you understand how we got it? This is the pH range when the relative activity was 60 for this enzyme. You're all with me?
Yes.
So we got this from 6.8 to 8.4, around 8.4. Now 8 to 2. We want the temperature range at which there is at least 60% activity. Again, we will go to which graph, Noor? Temperature or pH graph?
For the temperature? Temperature.
Yes, temperature. And then we will look at A22. So we'll go to the temperature graph, and the A22 is the graph that is X. This is A22. So we don't get confused. And we want the relative activity at 60. So I'll go to 60, I'll move horizontally until the graph stops me here. It stopped me here. And then the range will be from here till here. And you get values. This is the temperature range. This is around 31, 2, 3, 4, 5, 6, 7. This is 37. And this is 51, 2, 3, 4, 5, 6. It's not easy to count here on the screen. So it's nearly from 37 to 56 temperature. Let's look at them. We are right? Okay, correct. Now, and P3 is already given. Okay, so you just want to make sure that you can understand the graph. Keratin with a relative activity of at least 60% that has the widest working range of temperature. Mariam, I will look at which graph, temperature or pH?
Temperature.
Now, which one has the widest working range?
What
The
does
widest graph.
what does
The working range means width. The widest working range of temperature at 60%. So you will go at 60%. Okay, and you will look for the enzyme that has the widest width, the widest temperature range. Look, this looks very white to me. Look here.
I think it's
Where is
P3.
it? Yeah. Okay. Where does it continue? They fuse together. Oh yes, it continues here and then it goes here. So this is the widest at 60. It ranges from here until all the way to here. So it's P3 in my opinion. This is the widest temperature range, P3. Now the widest working range of pH. Adham, which graph will you look at, temperature or pH?
pH.
And let's go at 60 and look which one has the widest range. So this is 60%. Which graph has the widest range, do you think?
The one with the X's.
Yes, it's correct. One with the axis has the widest range, and the one is the axis is the A22. A22 enzyme. Let's see if we are wrong or we are right or what we are doing. P3 and A22. Very good. So we're doing a good job. Okay. Some detergents contain proteases to remove stains from clothes. These enzymes, they have a high relative activity in alkaline conditions. The scientists reported that K12 and A22 could be suitable. So these two enzymes, they are suitable for use in the detergent industry. With reference to figure 2.1 and figure 2.2, discuss the advantages and disadvantages, oh my goodness, of using K1 and A22 in the detergent industry. Thank God this question is only 3 marks. Then what would I do if I'm in place? I'm going to break down this question so that I don't miss any part. I'm going to discuss K12 and I'm going to discuss A22. What do they want? They want us to refer to the figure at the table and discuss the advantages and disadvantages of using these two enzymes. So this enzyme, I will discuss the advantages and the disadvantages. And the same for A22. I will discuss its advantages and its disadvantages. And I will go to the graph and I will try to get information about the advantage and the disadvantage for every enzyme. Let's just read it again. Some detergents contain protease to remove stains. These enzymes have a relative high activity in alkaline conditions. So we want to see which one can work in alkaline conditions. Scientists reported that K12 and A22 could be suitable. Okay, so an advantage if it works in alkaline conditions. Put this in mind. So let's go to K12. You will have to do that, whether you're like, yes, it's very boring. K12 is the one with a solid circle. Let's look at the temperature range. Let's look for advantages and disadvantages. Mariam, what's the temperature range for K12? K12 is the solid. What is the temperature range? It works at relatively low temperature. And then as the temperature increases, the relative activity increases. What is its peak temperature?
54, 55.
Around 50 something, which is not very high, by the way. This is a relatively low temperature. And after that, it will denature. So this can be a disadvantage. So the advantage is that it can work with an efficiency of 40% at low temperatures. Its peak temperature is 55, and after that, it will denature. So it will denature at relatively low temperatures, which I see, or you can write it as a disadvantage. Okay? So the advantage, it can work at low temperatures, but it will denature quickly at relatively also low temperatures. 50 to 70 is considered relatively a low temperature. Now let's look at its pH.
Doctor,
Yes.
can't we say as an advantage, K12, it's easy to reach its maximum activity at low temperatures?
Yes, you can say that because this is a suggest question. They want your opinion. And as long as you're getting correct information from the graph, it is correct. So this is an opinion. You think that it will quickly reach its optimum temperature. Yes, another student can think that the enzyme will denature quickly because the temperature is low. It's also correct, as long as you're giving correct values. So you understand the idea of the question. They want to see if you will extract correct information from the graph. This is called the skill question. You understand?
Yes.
Okay. So both suggestions or opinions, as long as you are getting correct information, yes, it's correct. Now let's look at the pH range. Now again, we're looking at this graph, the one with a solid line. The relative activity, the pH, what will you say here? Can it work well in alkaline conditions?
Not really.
The peak pH is around 8, so it can work well in relatively alkaline conditions, but not strongly alkaline. If it is strongly alkaline, it will denature. Okay? Now, you can write that. So the advantage, it can work well in a pH of 8, which is alkaline, but too alkaline, it will denature very quickly at a pH of 9. So you can do it like this. Now, A22, let's focus on A22 and give at least one advantage and one disadvantage. You understand how you will organize your answer? Because what really you get from the graphs is not as important as including all the information. You would never think of something like this, and your answers will get very messy, and you will miss advantage and disadvantage for every enzyme. Adam,
Yes, miss.
your handwriting has to improve. Has to improve. Because the
Fine.
examiner will not be able to read it. Okay?
Fine.
Now let's look at A22. A22 is X. So let's look at this graph, Adham. What can you say? And give me one advantage and one disadvantage when it comes to temperature.
A22?
Yes, the graph with X. Can it work at low temperature?
Yes.
It can. Both of them can work at low temperature. And then the activity will increase as the temperature rises. What is the optimum temperature?
About 45 degrees Celsius.
Yes, and you can say, like Maryam said, that the optimum temperature is quickly reached. And then after that, it will denature. Where is the graph? And actually, it will not denature quickly. This is an advantage because even at 80 degrees Celsius, there is a relative activity, but it is low. No, it will denature. It will denature at 70, which is relatively a high temperature. So it will not denature quickly. Okay?
Okay.
So it will work at low temperature, but of course, its relative activity will be low. The optimum will be around 45, which is easy to reach. And after that, it will denature. And it will completely denature at around 60, 70 here, which in my opinion is also low for an industrial enzyme. So this can be a disadvantage. Fine. Now let's look at pH. This is 82, which is the graph in axis. Can it work in alkaline conditions? What is the optimum?
It can, but it works at a neutral pH more.
Yes, so this is the disadvantage. So its optimum is neutral, not alkaline. And in alkaline conditions, the relative activity will start to denature. Okay?
Yes.
So if we look at the mark scheme, this is the outline that I wanted you to understand, that you will write the advantage and the disadvantage for every enzyme. Okay? An advantage and disadvantage for every enzyme. And they give you all options. So as long as you are retrieving correct information from the graph and you think it's an advantage or a disadvantage, you will get a grade. The idea is writing the answer for every enzyme. This is enzyme 1, advantage, disadvantage. Enzyme 2, advantage, disadvantage. That's why I keep on telling you, organize your answer. Organize your answer. It should be nice and clear and include everything. And for every enzyme, you have to include pH information about the pH and the temperature, and pH and the relative activity, and the temperature and the relative activity. Okay, so we want to learn together how to organize our answers. 3. The liver receives blood from the hepatic artery and from the hepatic portal vein. The hepatic portal vein transports blood from the digestive system. Hepatocytes are the main cell type of the liver. They have a wide range of functions, including synthesis of triglycerides and plasma proteins, detoxifying waste, energy. The hepatic artery branches from the main artery that transports blood from the heart. Name the main artery, Noor. What's the main artery? This is very easy. Noor, where did you go? Hello?
Yes. Yes.
What is the main artery, Noor,
Aorta.
that transports blood? Huh?
The
What is the
transports
main
blood.
artery? To
الأورta.
all the organs. Aorta, yes, I'm waiting for the answer. The aorta. Good. Blood arriving at the liver enters specialized blood vessels known as sinusoids. Figure 3.1 is a diagram of part of a sinusoid and surrounding hepatocytes. A second type of cell found in the liver, a Kupffer cell, is also shown. Kupffer cells are phagocytic cells of the immune system. Okay, this is the Kupffer cell. It looks very much like a neutrophil. So just one advantage of having large gaps between the endothelial cells of the sinusoid, as shown in figure 3.1. another suggest question, but it's easy. Why do we have here large gaps, Adham, between the endothelial cells of the sinusoid?
For the cover cells to squeeze in.
Very good, for the cover cell to go out and reach the tissue fluid or site of infection. Good. In addition to removing bacteria present in the blood inside the sinusoid, the cover cells are also able to remove old or damaged red blood cells. Describe the mode of action of a cover cell in removing and breaking down a damaged red blood cell. 4 marks. What will we discuss in detail here, Mariam?
The hydrolytic enzymes endocytosis.
What?
The enzymes that
hydrolytic-
break down the damaged cells and organs.
No, this is part of the answer. Describe the mode of action of the cover cell in removing damaged red blood cells and removing something foreign. This cover cell, what does it look like to you? And they said they are phagocytic cells. So what will we discuss in detail? All the steps of what?
Phagocytosis.
Yes, all the steps of phagocytosis. And the last step of phagocytosis will be the release of the hydrolytic enzymes. So the phagocytosis, you will start with all the steps. You know this long column in your notes. It will be attracted to the red blood cell because the red blood cell will either release a histamine or it will be marked by an antibody. And then after it is attracted to the red blood cell, what will it do? So first step is attraction, chemotaxis, if you remember, and then what will it do after the cover cell reaches the red blood cell? It will do what, Mariam? You said the word. Endo? Phago?
Endocytosis.
Yes, it will swallow it. It will do the phagocytosis, so it will go to it, and then it will engulf. It will stretch out or extend out its cell membrane. It will do engulfing, or it will do the phagocytosis, or it will do the swallowing. And by the way, when the cell membranes reach out, this is called pseudopodia. You want to know? And then after it will swallow it, it will put it in a
Vacuum.
what's the name of this vesicle? It's called the
Phagosome.
yes, the phagosome. This vesicle. And who will come and fuse with the phagosome?
Lysosome.
Correct, the lysosome. And when the lysosome fuse with the phagosome, they will create a what?
Phagolysosome.
Correct, phagolysosome. And then it will be the last step, which will be the
Digestion breaking down.
very good, using which enzymes?
Hydrolytic enzymes.
Correct, and you can mention a few examples: proteases, the nucleases that will break down the phosphodiester bonds, the any type of enzyme, glycosidic bonds, like carbohydrates that will break down the glycosidic bond, the lipases, all the any example of any hydrolytic enzyme. Okay. So you will mention all the steps of phagocytosis. Let's look at the marks in case we're not right. So where are we? Water. We answered this question. Endocytosis, phagocytosis, the red blood cell will be engulfed very by the pseudopodia, correct? And then the phagosome, and then the lysosome will fuse, and the lysosome will contain hydrolytic enzymes. I'll give examples: digestion to produce and then we will, any valid point, example, suggestion, or detection, which we said at the very beginning. So we said, or we gave the answer in full. The question that does not finish. Figure 3.2 is a transmission electron micrograph. If you notice, they always get questions over the transmission in every variant. Transmission, this means we're looking inside the cell. So we have to be looking at a particular organelle, not the whole cell, of part of a hepatocyte showing some cell structures. The peroxisome shown in figure 3.2 is a spherical organelle bound by a single membrane. It carries out a variety of enzyme-catalyzed metabolic reactions, including detoxification. Some of these reactions require oxygen. So this is the peroxisome, and we have here organelles. What's this organelle? It's very obvious. Noor.
Mitochondria.
Yes, it's a mitochondria. And by the way, this is also a mitochondria, but this is just a different cross section. And how did you know that it's a mitochondria?
الشيب.
Yes, and they have internal foldings called what?
الانترنال فورس كريستي او حاجه كده
Yes, okay. They increase the surface area for aerobic respiration. Okay, let's see what do these people want. Describe the evidence visible in figure 3.2 apart from the presence of a peroxisome that indicates some of the functions of the hepatocyte. Okay, this is 3 months. So we want describe the evidence visible. Add labels to figure 3.2 to identify the location of any cell structure, if not already labeled, that are part of your evidence. Okay, now, what can you label? We just did. What can you label other than the mitochondria? What can you see that can be labeled? What's this? So they want you to label organelles and discuss their function in the peroxisome. They told us that the peroxisome has several functions. It carries out a variety of enzyme-catalyzed metabolic reactions, including detoxification, and some of these reactions require oxygen. And here, before all that, they gave us a list, I remember, of the functions of the liver cells. The synthesis of triglycerides and plasma proteins, and breaking down waste and storing energy. So we want to label everything here and discuss its function. We said mitochondria. What else?
There's an endoplasmic reticulum.
Yes, it's very obvious here. This is the rough or the smooth?
There is rough and there is smooth. Some of them are present.
Correct. This is obviously the rough endoplasmic reticulum. As long as there is rough endoplasmic reticulum, there will have to be smooth endoplasmic reticulum. Maybe the smooth is not very clear, like these structures, faint structures, because they don't have ribosomes on their surface and they are smooth, so they're not very obvious. So, but as long as you see a rough endoplasmic reticulum, there will have to be a smooth endoplasmic reticulum. And this is mitochondria, and this is another mitochondria. This is the peroxisome. These are the glycogen granules. What is the function of the glycogen granules? What do you think? What do they store? Come on.
Glucose.
Yes, they store carbohydrates. They store glycogen. This is the liver, okay? So this is for storage. And what is the function of the lipid droplets?
For energy.
They store lipids. And these lipids can be used for other things. These lipids can be used for energy, or they can be used for the making of the triglycerides and plasma protein. I want you just not to forget the question. At the beginning of the question, they give you very important information. They are talking about a particular organelle, and this organelle, it does two things. This hepatocyte said it does two things. It will produce triglycerides and plasma proteins, so it must have the organelles to do that. And it will also break down waste, so it has the buffer cell, and it stores energy. So that is why it has the glycogen granule. So it is a multi-purpose cell. Do you understand? This hepatocyte is the main cell type of the liver. It does a lot of things. So it must have the organelles to help it do a lot of things. It synthesizes triglycerides and plasma proteins. Adham, which organelles do you see here that will help the hepatocyte to produce triglycerides and plasma proteins?
Golgi body and smooth endoplasmic reticulum.
We did not say there is a Golgi body, but of course it does have one, but we don't see it in the picture. We said it has
See it.
a smooth
I really
endoplasmic
see it.
reticulum. Yes, and we said it has a rough endoplasmic reticulum. The smooth endoplasmic reticulum is used in the making of what?
Lipids.
Yes, triglycerides. So it is used in the making of triglycerides just like they said in the question, or lipids, correct? And the rough endoplasmic reticulum is needed to make what?
Proteins.
Very good. It is needed to make the proteins, for the modification of proteins, for the synthesis, whatever. Okay. Now,
بص هو
we have
تقريبا
other
اللي على اليمين
organelles.
هو تقريبا اللي على اليمين بتاع الصورة ده جولجي بادي واخد كيرف كده.
خلاص, if you can see it labeled, I don't think they will consider it as wrong. Okay.
اوكي.
I know which one you're talking about. Now,
يعني
the
مش هيعملوا ريجكت او حاجة؟
mitochondria. The mitochondria, excuse me?
مش هيعملوا ريجكت او حاجة زي كده؟
لا, مش هيعملوا عشان هو شبه الاندوبلازم reticulum. They are made up of flattened sacs. They're just not fused. Now the mitochondria. Now this is obvious. Okay, the mitochondria, what will you write as a function?
Mitochondria synthesizes ATP using oxygen.
Very good. So it produces ATP in aerobic respiration. And of course, this is needed in all the chemical reactions inside the cell for the making of proteins, triglycerides, whatever. Now, we also have other organelles that are already labeled. The glycogen granules and the lipid droplets. Glycogen granules,
Stores
they
energy.
store what? Stores.
Carbohydrates which are used to make energy.
Yes, glycogen, they can be used in respiration, broken down. And the lipid granules, they store
Even more energy.
They store lipids. Okay, so this is out of 3 marks only. If you label only the mitochondria and the rough endoplasmic reticulum and you discuss their functions, you will take the grade. Okay, in addition to the labels that are already given, because again, you read the question carefully, add labels to identify the location of any cell structures, if not already labeled, that are part of your evidence. So you can add labels or you can use labels that are already there. So we added 3 labels, the smooth and the rough and the mitochondria. If you don't see the smooth and you only label the rough and the mitochondria and discuss the other 2, you will get the 3 out of 3. Okay? The mitochondria in figure 3.2 are larger than the peroxisome. State one other difference visible in figure 3.2 between a peroxisome and a mitochondria. Noor, what do you see difference between a peroxisome and the mitochondria?
The mitochondria has the folding. Christy.
Yes, you are right. This can be a difference. It does have inner foldings called crista that increase the surface area for aerobic respiration. I want you to give me another difference, something that you can see and something that we look in the lesson.
The mitochondria has a double membrane and the peroxisome has one membrane.
Yes. And actually, it was also said in part of the question, I remember, yes, bound by a single membrane. Be very smart. They are very tricky. Like they give you the information in the question. They told you that the peroxisome is bound by a single membrane. And we know, even if you don't see it, that organelles like the mitochondria and the nuclear membrane and the plasma membrane and the chloroplast in plants, they are surrounded by a double membrane. Okay?
Okay.
So when you put the mitochondria, yes, they have crista. I will not argue here. And they also have a double membrane. Some of the enzymes used within mitochondria can be synthesized by the organelle. Peroxisomes cannot synthesize any of the enzymes that they contain. So just why a mitochondria can synthesize enzymes, but a peroxisome cannot synthesize enzymes. Again, this is the lesson cell structure. Adam, why can mitochondria produce enzymes, but the peroxisome cannot?
Because it has circular DNA inside of it.
Correct. What else? Because it does have circular DNA.
And 70S ribosomes.
Excellent. It does have 70 ribosomes, and this DNA and the ribosomes can be used in what?
Translation.
Transcription and translation in protein synthesis. Yes?
Okay.
The DNA, I can use it to copy and make the messenger RNA, and the ribosome for the protein synthesis, transcription, and translation. Very good. The peroxisome does not have that. And also, they have all the enzymes needed in all the steps for protein synthesis. We need RNA polymerase, we need the transfer RNA molecule, we need everything is available in the mitochondria. One of the enzymes present in the peroxisomes is the catalase enzyme. This enzyme catalyzes the breakdown of hydrogen peroxide to harmless products. Suggest question. Why is it useful to the cell for this reaction to take place within the peroxisome? Why is it an advantage that this enzyme is inside the peroxisome and it is isolated? And why? I want to give me 2 reasons. Mariam, yeah, come on, speak up. Why is it an advantage that this enzyme and hydrogen peroxide is inside the peroxisome and not outside the peroxisome?
To not damage the rest of the organelles.
So the what?
To not damage the rest of the organelles.
Yes, correct. So the hydrogen peroxide is isolated in one place and it is toxic. The hydrogen peroxide is toxic, so it doesn't damage the remaining organelles. Yes, it's that simple. And the enzyme is localized in one place. If you remember, this is one of the functions of the cell membrane. It's called compartmentalization. We put it in one compartment so that it is in one place, it is isolated, localized, and the waste products do not harm the rest of the cell. 2 more questions. These paper 2 is always 6 questions. The alveoli of the lungs are the main gas exchange surface. Explain how blood flow through the alveoli capillaries help to maintain a steep diffusion gradient for gas exchange. This is an easy question only, also. Adam,
Yes, doctor.
can you tell me how the blood flow will maintain steep diffusion gradient?
The capillaries, the blood capillaries are very close to the alveolar surface, which shortens the diffusion distance of gases to the blood.
No.
No?
We're talking about the diffusion gradient, difference in concentration between the gases. What is the gas that is found in the capillaries? Mainly, it is bringing which gas from the body?
Carbon dioxide.
Yes. So the capillaries, they have a high concentration of carbon dioxide.
Okay.
And the alveoli contains a high concentration of what? Which gas?
Oxygen.
Yes. So the capillaries arriving from the body, they are deoxygenated. They have a high concentration of carbon dioxide, and there is less carbon dioxide in the alveoli. So this will create a great concentration gradient, and the carbon dioxide will diffuse out of the capillaries quickly so that they go out of the lungs and they are exhaled. And the oxygen, it is in high concentration in the alveoli, and it is in low concentration in the capillaries. So this will also create a high concentration difference or gradient, and the oxygen will diffuse quickly from the alveoli into the capillaries, and the capillaries will take it away to the body. Okay, this question is common, by the way.
Okay.
So you will mention that the blood arriving at the lungs, it is deoxygenated blood with high CO2. I have low CO2 in the alveoli. This creates a concentration gradient that will make the diffusion of CO2 out of the blood capillaries quickly. And you will discuss oxygen. Oxygen is in high concentration in the alveoli and in low concentration in the capillaries. High concentration in the alveoli, low concentration in the capillaries, also greater concentration gradient and faster diffusion of oxygen from the alveoli into the capillaries that will take it away. Okay?
Okay.
Okay. Mariam, are you there?
Yes, doctor.
Okay. Explain the role of the elastic fibers in the alveolar wall during ventilation. Direct question. How do the elastic fibers help in ventilation?
Elastic fiber, they allow the contraction or okay, it allows the expanding and stretching of the stretching.
Yes, you're right. It does something during inhalation. When you inhale, what happens to your alveoli? Do they stretch or they recoil?
During inhalation, it stretches.
Very good, because it is filled with air. What if the elastic fibers were not there? It will overstretch and it will
Burst.
Burst. So during inhalation, the alveoli will stretch, but the elastic fibers, they are important because they prevent overstretching. Clear? Now,
Yes.
during exhalation, what does the elastic fibers do?
They allow the alveoli to recoil.
Very good. They squeeze the air out. They are squeezing the air out. If the elastic fibers were not there, the alveoli will not be able to recoil, and the air will also be trapped, and it will result in bursting of the alveoli. So the elastic fibers, they allow stretch, they allow recoil, stretch when you inhale, but prevent overstretching so the alveoli does not burst, and they allow the recoiling when you exhale to push out the air. If recoiling was not possible, the air will be trapped and the alveoli will burst. So in both cases, if it was not there, it will burst. That is why it is very important. Here, again, this is an important question, even though it's easy, but I'll highlight it for you. It's an important question. And this one is important, even though it is easy also, but I don't know why you don't get it easily. So let's highlight it. Okay, some cells in the alveolar wall are specialized to secrete a surfactant to prevent collapse of the alveoli at the end of the exhalation. In these cells, surfactant is stored in membrane-bound organelles known as lamellar bodies. Surfactant is a mixture of lipids, mainly phospholipids and some proteins. A protein known as ATP binding cassette transporter A3 is needed to move a surfactant phospholipid into the lamellar bodies from the surrounding systole, or the fluid part of the cytoplasm. Suggest and explain the features of a protein ABCA3 that makes it suited to its function. My goodness. Okay, this protein, it is known as an ATP binding cassette transporter. Okay, it's needed to move. So this surfactant will not move across the cell membrane except if this protein is present. So this protein is needed to move the surfactant into the lamellar bodies from the surrounding fluid. So in other words, I have here a surrounding. My pen doesn't work anymore. So I have a surrounding, and I want the surfactant to move in, and it will not be able to move in except when this protein is present. So what is this protein? And it is an ATP binding protein. This is which type of protein? And where is it found? No, give me an answer.
Channel protein.
It's close, you're very close. It is a protein in the cell membrane. But is it a channel protein or a carrier protein?
Carrier.
Yes, carrier. Why is it a carrier protein? Because they said here it binds to ATP. So it is an ATP, it has an associated ATPase enzyme. You took it in A2, yes? What is the function of the ATPase enzyme? It binds to ATP even if you did not take it in A2. What does it produce?
ADP and PAR.
ADP plus PI.
Very good. So it is going to produce energy. So this is a channel protein or a carrier protein?
It's a carrier protein.
Correct. If you write as a channel protein, you will get half the grade, or you will get a grade. It's not totally wrong. But this is a protein that is found in the cell membrane, excuse me. And they said that it is known as ATP binding cassette, so it is associated with ATP. So it is a carrier protein. Now discuss all the features of this carrier protein. We took it in detail in the cell membrane. So this is a carrier protein. What is the function of the carrier protein? What does it do? It helps in what?
Transport.
Yes, transport of what? And from what concentration to what concentration? What's the name of the process?
Active transport.
Facilitated diffusion.
Yes, active transport. This active transport will carry molecules from where to where?
High concentration to low concentration.
Ya Rabbi, ya Rabbi, ya Rabbi.
Low to high, low to high.
Low to
From
high.
low
Sorry.
to high. From low to high. And it needs energy. That is why it is bound to ATPase, or it is known as ATP binding, because it will need energy. Now, this energy is needed for what? Why do I need energy? To change what? Mariam, help. Doesn't it change shape? Carrier proteins, don't they carry materials from low to high concentration? They change shape. And what is the name of this change of shape?
Conformational changes.
Thank you. Conformational changes. Okay, please study well. Please study every single detail. Okay, now these carrier proteins, they transport. What is the nature of the molecules that they transport? Molecules that are soluble in the phospholipid bilayer, or they are not soluble in the phospholipid bilayer? They cannot, they can or they cannot cross. They cannot cross the phospholipid bilayer because most probably they are hydrophilic. These carrier proteins, their internal side or their interior is hydrophilic, so that they can accept hydrophilic or polar molecules. Okay, so you will discuss everything about carrier protein. But my problem is, I doubt that you will even realize that it's a carrier protein. Now, this is a twisted question. I call it a twisted question. They can easily ask you, what are the features of a carrier protein? But they don't want to do that. They want to give you a hard time. They are not nice people. Okay? They gave you a story and they wanted you to think of which type of protein is it and discuss its characteristics. And the problem is here. You know what is a carrier protein and you studied it. But the problem is that you should reach the conclusion that it is a carrier protein. So you will get a grade if you will mention it is any type of protein that will carry materials across the cell membrane. But because it is ATP binding and associated with ATP synthase, it is a carrier protein. And this is something that I doubt you will know on your own. They are very cruel. So let's look at the mark scheme. This is number 4C. Do you know that it is a protein? Adam did tell me it's a protein, but I think he told me channel. Okay. It's stated as facilitated diffusion allow only maximum 1 mark, not the 3 marks. So any three fronts. It's a carrier or transport protein. You will be given a grade mentioning that it is a protein, but it's a carrier protein. Okay? Reject channel protein. It's not a channel protein. It is a carrier protein, like we said. It has a specific binding site for the phospholipid. ATP is used. It is hydrolyzed. It is needed for active transport to provide energy. Okay? We'll move against a concentration gradient. We said all that. We talked about the conformational change. Okay? So will you get it that it is a carrier protein, a specific one? This is my question. So I'm going to highlight this question for you also. I tell you, please read the question carefully because they give you hints in the question. They give you the answer in the question. Okay. The gene ABCA3 codes for this protein, and the gene is 80 KB or 80,000 base pairs long, and it is composed of introns and exons. Protein ABCA3 is composed of 1704 amino acids. Figure 4.1 shows the flow of genetic information in the production of this protein. So the gene will go through X and then it will create the primary RNA. What is X?
Transcription.
Correct. So X is transcription. You know it. And then the messenger RNA will create the polypeptide. So what is Y?
Translation.
Yes, this is easy. You never get straightforward questions, unfortunately. Really. Okay. A triplet of bases codes for one amino acid. This fact only partially explains how the activity of the gene, which is 80 base pairs long, can result in the protein AB, I don't know what, which is only 1704 amino acids long. Now, this was what I was talking about yesterday when I told you that this question became very common in the past years. Again, another suggest question. Suggest questions are tricky ones. Suggest other reasons to explain the difference in the number of base pairs in the gene compared with the number of amino acids in the protein. So they're telling you that the number of base pairs in the gene is much more than the number of the amino acids in the protein. I want 3 reasons why. I gave you the answer yesterday in the WhatsApp. Why do I have lots of base pairs, but in the end, the final protein is much shorter? Mariam, can you give me an answer?
Yes, the start and the stop codons are removed.
Yes, base pairs code for the start codons and the stop codon and the start and the stop codons. They will be finally removed from the final protein. What else?
During splicing, the non-coding genes, the introns, are removed.
Yes, some base sequences are not coding. We have lots of base sequences, but not all of them code for amino acids. Some of them are non-coding and they're called introns. And these are removed during the splicing before translation is made. Okay. Give me one last reason.
The genes are degenerate, so more than one base triplet can code for the same amino acid.
Very good. So sometimes they are just repetitive, or they're different, but they call for the same amino acid. Okay, name the species of bacterium causing the transmission of TB from dairy cows to humans. What's the other?
Mycobacterium bovis.
Bovis.
Yes, the bovis one, the bovis. Okay. Outline a control measure that can be taken to protect people that consume milk and dairy products from these infected cattle. How can you protect yourself from infected milk and infected meat? This is also easy.
Pasteurization.
Yes, very good. Heated pasteurization, cook well and heat and pasteurize. Cattle are not usually affected by the tuberculosis, but the pathogen can cause disease in other animals. A few cases of transmission of TB from people to animals have been reported. Explain the most likely mode of transmission of TB from an infected person to an animal. 2 marks.
Infected person coughs. So
Yes, you are correct.
the bacteria is breathed in by the animal.
Okay, but there is a word here. This bacteria, what is the mode of transmission of this bacteria? Bloodborne or airborne or what?
Airborne.
Airborne droplet
Yes.
infection.
So this bacteria is airborne. It is found in infected droplets, in aerosol, in the air, in other words. So what you said is correct. Okay, you will cough, you will sneeze in front of the plant, I mean the animal, and it will become sick. Okay. Outline the treatment that is used to kill the Mycobacterium tuberculosis and latent TB infections. Again, this is easy. How do we kill this type of infection? One easy question, finally. Antibiotic, okay? But
They
this
use
is 2 marks.
They use 4 antibiotics, and for the second mark, isolation.
Okay, yes, there are 2 marks. So antibiotic alone is not enough. You can, to make sure that you will kill this TB, this is a very strong bacteria, you will use more than one type of antibiotic. You will use it for a long time, the correct dose. You can mention one mode of action of one of the antibiotics that you studied. We studied the penicillin only. The penicillin contains these, if you remember, it's going to inhibit the transferase enzyme that will prevent the cross-links in the making of the cell wall of the bacteria while the bacteria is growing. So just give another piece of information because it's 2 marks. It's enough to say that I will use more than one combination of antibiotics to make sure that I kill all the bacteria or for a long period of time. Actually, TB is a very strong bacteria and it needs a long time to treat. Okay. Now, M. tuberculosis can spread in the blood and lymph to other organs in the body. In very rare cases, a disease known as mycotic aneurysm can be caused by infection of the arterial wall, particularly in elastic arteries. The damage caused by the pathogen can lead to a rupture, bursting the artery, with reference to the structure of the wall of elastic arteries. Suggest how damage caused by the M. tuberculosis infection can lead to the rupture of the artery. You may draw a diagram of if you wish. So here, what diagram will you do? And you have a space for the diagram.
We will draw
What will you
the
do?
layers of an artery.
Of course, you will draw the elastic artery and the different layers of the artery. Now, an elastic artery, why is it called elastic? Because what is present?
It
Elastic
has a lot
fibers.
of elastic fibers.
Very, very good. And the elastic fibers are present in which layer?
Tunica media.
Media.
Yes, correct. So you will space for the diagram. When I come to draw an artery, I will draw a wide lumen or a narrow lumen.
Narrow lumen.
Arrow.
Correct. So I will draw a narrow lumen. So I'll draw something like this, not this. Okay. So it's a narrow lumen and it is lined by the tunica. Remind me,
Intim.
the tunica intima, made up of a thin layer of endothelial or cuboidal cells. Okay. And then a thick layer of tunica,
Media.
the tunica media, what contains mainly, in this case, mainly what?
Elastic fibers.
Mainly elastic fibers. It does contain, of course, little smooth muscle and little collagen, but mainly elastic fibers. And then I have the tunica,
External.
the last layer is the tunica, what very good externa, that will have mainly collagen. No elastic fibers and no smooth muscles in the tunica externa. All of these tissues are found in the tunica media. But in that case, because it is an elastic artery, it will contain mainly elastic fibers. And this is the tunica intima. This is a small diagram here. Now, you will discuss the three layers. And then, so you will discuss the three layers in a nice organized order. You will say tunica intima. A thin layer of cuboidal cells, smooth epithelial layer, less friction so that the blood flows through smoothly, and we'll talk about it. Tunica media, the thickest layer, which contains mainly elastic fibers, and then the tunica adventitia or externa, that contains mainly collagen for support, so that it doesn't burst, so it doesn't rupture, and it connects with the surrounding tissues. Now, tunica media. What happens? Let's answer the question. Guess how the damage will cause the artery. If the elastic fibers are damaged, what will happen?
To rupture.
Okay, I love elaboration. Okay, what is the function of any elastic fiber? Like the alveoli, elastic fibers allow the artery to what?
Stretch and recoil.
Very good. I want you to put in mind that the smooth muscle, it's a muscle. Muscles, we describe their function by contraction and relaxation to control the diameter. This is an artery that has many elastic fibers, so it can stretch and recoil. Now, why is it important that it stretches when the blood is flowing?
So that
You just said it.
it doesn't rupture.
Yes, because this blood is under
High pressure.
very good, and we don't want it to rupture, so that it will smooth out a pulsatile flow, if you remember. So the elastic fibers, when they are damaged, it will not be able to stretch and recoil, and it will not be able to withstand the high pressure, and it will rupture. So this is right. But what I assume, knowing you, is that you go directly and discuss what will happen to the artery when the elastic fibers are damaged. You will not draw. You will not talk about the different layers. You will not give a thorough, nice, complete answer. You always underwrite. Okay? When there are no elastic fibers, it will not stretch, it will not recoil. It will overstretch and it will rupture, and it will not recoil, and the blood will become stagnant. So elastic fibers, don't forget, they stretch. Normally, they allow stretching and recoiling. No elastic fibers, there will be no stretch and no recoil, or it will overstretch and it will not be able to recoil. Clear?
Clear.
Yes.
Okay. Very good. Now the last question. Finally, the last question. Water that is absorbed from the soil solution by the roots of a plant enters xylem vessels and is transported to the leaves and the buds. Figure 6.1 shows 4 important requirements for the efficient transport of water from the roots to the leaves. What are these requirements? Tension needs to be created in the xylem vessels. Xylem vessels must be able to cope with the tension created by the movement of water. Requirements for the efficient transport: there should be minimum resistance to the flow of water, and water in xylem vessels must be in continuous unbroken columns. Okay. Name the specialized cells that are arranged end to end to form xylem vessels. This is also easy. What's
Vessel
the name
element.
of the vessel elements? Yes, okay, the vessel elements. Explain how tension is created in the xylem vessels. Why is there tension created?
Because of the transpiration pull.
Correct. Now, how is that? Describe. Don't just write it and leave. Now, what happens at the transpiration pull? Why is there a pull anyway? Why is there a pull that will pull the water from the soil to the leaves?
Because of cohesion and adhesion.
No.
No?
No.
Right.
Due to the changes in the
I'm talking about the tension. Yes, due to the changes in pressure. You can write cohesion and adhesion, yes, but this is not the reason why the tension is there. There is a tension because I have high hydrostatic pressure in the soil. And at the leaves, because of the transpiration, what do I have? Low or high hydrostatic pressure?
Low hydrostatic pressure.
Low.
Low hydrostatic pressure. And this changes in pressure will create tension, will create the transpiration pull, will pull the water. And also cohesion and adhesion forces will help. But the tension is created, the negative pressure is created due to the changes in pressure. And this will make the xylem vessel to move inwards. And if the xylem was not strong, the xylem will collapse. But does it collapse?
No.
No. Why does it not collapse?
Contains lignin.
Very good. Okay. And also you can mention cohesion and adhesion, of course. Fine. So you will mention everything here to be on the safe side, even if it's too much. Know what you write.
Understood.
Walls of the cells forming the xylem vessels become lignified during development. Explain how this feature is important for the efficient transport of water. We just said it. This lignin, why is it important?
Prevents bursting.
It's not bursting. When there is
Collapse.
negative pressure, it will collapse inward. There is inward pressure. Will this happen? It will prevent what?
Prevent
This will
collapse.
burst. It will collapse. Yes, it will prevent inward collapse. So it will prevent collapse. It will provide strength.
And support.
It will provide support. But can we think of anything? Also, the lignin will help in the adhesion of water. The water moves up the xylem. Okay. It will withstand the negative pressure that is created inside the xylem. I just want to write everything I'm thinking of. Okay. Now, during the development of the xylem, the end walls of the cells forming the vessels break down. This contributes to minimizing resistance to the flow of water. Describe one other main change that needs to occur to these cells so that their structure becomes suitable. Now, this is easy. How is the xylem suitable for the flow of water? Come on.
It's hollow.
It's hollow. No nucleus, no cytoplasm,
No organelles.
no organelles. It has lateral pits for the lateral movement of water. Don't forget, plasmodesmata is in the phloem. It's empty. The exam is finished, finally. Okay. Please solve lots of exams because every exam teaches you something. We did not look at the mark schemes. I'll send you this exam. And if you have any reservations, then okay. The tricky questions are usually the suggest questions. I highlighted the tricky questions for you if you want to revise anything. Now we will have Wednesday will be late. So we cannot have a class on Wednesday. We'll have a class on Monday only. That's why I had a class today. Okay?
Okay.
See you. Please study very, very, very well. And tell me if you have any questions.
Okay.
Bye.
Bye.
Understood.